Wright. We're from the University of Sheffield's Institute for Sustainable Food. And our project is looking at harnessing and integrating suppressive microbes found in soils and then deploying them in a synthetic community context into synthetic soils in low input horticulture, which I'm going to glaze over that very quickly. We kind of identified several problems as our sort of starting point. The first thing is what we're looking at, which is hydroponics. Why hydroponics? We've probably all heard the big headline things: 10% of the water that you would use in conventional agriculture, and you reduce nutrients and you can use them all year round. But what's more interesting to me is that the speculators forecast 8% compound growth on hydroponics up to 2025. And when you bring that back to the UK, for example, and the conventional agricultural challenges that we've got, like, for example, loss of fenland peats, we could see why demand will be increasing as we go forward. There's some significant drawbacks with hydroponics, and one that we've really focused on with this work is the lack of suppressive ability within these systems. So once you have a pathogen introduced to a system, they tend to be quite devastating to the whole system. And with that comes a significant costs in either treatment or defense in the first place. And a lot of this has to do with the fact that there is no real biological priming of defenses of the plants to start with. When they're brought up in hydroponic systems, they obviously don't have a natural ecosystem with which they can deal with any kind of invasion with a pathogen or a pest. The disease that we're targeted with this project is tomato root mat disease, which we know is primarily caused by Rhizobium Radiobacter. And the reason we're looking at that is that in the UK, our hydroponic tomato production is about 80% of our tomatoes produced in the UK are produced hydroponically, and most of the varieties, I think all but one, are susceptible to root mat disease. With that in mind, the AHDB has spent a bit of time looking into this over the last few years, and one of their big reports, which was completed in 2018, interviewed a number of producers. 88% of them had seen tomato root mat disease at some point, and up to 5% of their crops were affected by this. At the moment, the AHDB estimates this would cost £29,000 a hectare a year for nurseries for treatment. Treatment's a funny word to use, or control, which is obviously a significant amount and only likely to increase as the disease sort of spreads around the UK more. What they did do in their experimental work is explore single shots off-the-shelf biocontrol treatments, but they showed that these had little success in controlling root mat disease. And one interesting thing is that they did note within their hydroponic systems that the root mat disease had an affinity with some substrates, like natural substrates like coconut coir, for example. And it's important also to remember that tomato root mat disease is not just tomato related, it also affects cucumbers. And if we relate it to a closely related disease, which is called crown gall, we know that affects other cultivars as well, such as raspberries or roses. So potentially it could spread into other crops which have relevant effects for us in the UK. Just to give you an idea of what root mat disease looks like, I've got a couple of pictures here from the AHDB's work. One thing to note here is, I mean, obviously it causes a vast amount of root proliferation, but on the picture on your right also shows the mechanical issues related to hydroponics, where you might get roots ingressing into pipes and things like that as well, which also brings about its own costs. So the bacteria that's sort of at the root of all of this is, for want of a better phrase, is Radiobacter. And this is just a sort of life cycle shot of it. In order for the Radiobacter to cause root mat disease, it must have a plasmid which has a root-inducing gene on it. This plasmid is injected into the tomato's nucleotide DNA, and then that DNA will cause excess hormone production, which will then, as a result, cause excessive root formation for the plant. What's interesting about these roots and proliferation is that they also produce an exudate, which are low molecular weight molecules called opines, and these opines are used by the bacteria for catabolism. So it's effectively a food for the bacteria to thrive on. Just focusing in on Radiobacter a bit more. So Radiobacter is actually quite well characterized through the study of crown gall disease. Crown gall is where you get tumors forming on the foliar or stem parts of the plant, so above ground. And the difference between these is the plasmid that they have has a tumor-inducing component rather than a root-inducing component. The AHDB in their studies have looked at qPCR to confirm infection and to determine whether or not these plasmids were present. This process can be quite time-consuming and is time-sensitive on its own as well. But also, what was interesting about the AHDB work is, well, like I alluded to earlier, the single-shot biocontrol is not necessarily the best route, and in fact, it might need to be more of a system-wide biocontrol. The opine exudates are quite interesting to me personally, because I'd like to use them as a way of determining infection as well. And the reason for wanting another way and a faster, cheaper way of determining infection is you will see symptoms as much as 8 weeks after infection in the plants, which is not great. Obviously, the colonization is well and truly underway at 8 weeks. And in hydroponics especially, it's particularly hard to detect root mat disease because it's happening at the root zone. So it'll be happening within the substrate, and the substrates in hydroponics are normally wrapped in polythene sheets or are in guttering systems, so you might not see the root matting occurring. And the plant matter above ground is actually appears fairly unaffected until you get to the point of fruiting. So it can take a while to determine whether or not you actually have the disease when it may have been around for several weeks. This slide's just to sort of illustrate that point. So you can see on the left a very healthy plant. In the middle, the top surface of your mineral wool there doesn't appear to have any root matting. And then on the right, this is quite early on in our first patho system run, but we can see what looks to be the beginnings of root matting. So you can understand why we might want to detect it sooner rather than later. So our solution is to, we co-designed work going forward with this AHDB work and with the AHDB, and we kind of have a 3 work package system where we'd like to isolate and understand the mechanistic basis of disease suppressive microbes in soils, and then we would like to develop some foam-based horticultural substrate, which Harry's been working on his PhD, and then apply these suppressive microbes within these substrates. And then finally, we'll look to optimize what I've termed here as a synthetic soil with a synthetic community for commercial trials at a later point. We have been so far working in 2 parallel streams. So Harry's going to take over the first stream, and then I'll talk about what I've been working on.
Sure. Right. Hi, everyone. I'm Harry. I'm actually got a chemistry and chemical engineering background. My PhD was on developing polyurethane foams for use in hydroponics. Today I'm going to be talking with 2 kind of hats on. So I'm going to be talking about the microbiome and identifying disease suppressive soils. Our postdoc, who's kind of running this part of the project, is on maternity leave, so I'm taking over from her. I don't think I'll do it as much justice as she would, but bear with me. So the first part of the plan is to identify possible disease suppressive soils and then screen these to see if they induce or if they're capable of disease suppression, isolate and identify the microbial communities from these soils, and then transfer these communities to a synthetic substrate in hydroponics and see if any of the bacteria or what bacteria is actually able to survive this transfer process into a whole new environment, and see if they're actually able to continue suppressing disease in a hydroponic system. So the main thing what we're going to be looking at, is if you look at microbial control agents, they generally have a direct action or indirect action. Direct action would mean they actually kill a pathogen. Indirect action is more what we're expecting to see from disease suppressive soils, where they either act as competition for the pathogen, or they can do something called induced systemic resistance for the plant. which basically primes the plant's defense, which means if there is a pathogen attack, it's able to respond much stronger and much faster than an unprimed system that doesn't have this induced systemic resistance. So we're going to be hopefully finding soils that are able to provide ISR and then transfer that into a synthetic substrate. What Jake mentioned earlier about synthetic substrates and rock wool in particular, and single bacteria inoculants, which haven't worked that well against Rhizobium Radiobacter, sorry. And we kind of think there's a few reasons for this. Is one, the single strains aren't really suitable for hydroponics, so they're not used to that environment, and they're not doing well in that environment. But secondly is the actual substrate itself, and that the substrate perhaps doesn't allow for enough adhesion or for the bacteria to actually survive in the substrate. So these are some pictures of polyurethane foam that I developed during my PhD. But what's really interesting for this project is not so much how well it works from a growth perspective. It's more the fact that we can completely vary all the physical and chemical properties of the substrate. So these are some ranges I've got from previous work I've done where we can change density, cell size, water holding capacity, water drop penetration time, so how hydrophilic or hydrophobic the substrate is, the percentage of open cell in a foam, which is basically how easy roots can go through it, and also affects the water holding properties of the foam. But what we can really do with this, hopefully, is optimize a substrate around the bacterial life. So make it that the bacteria we want to colonize is actually able to colonize the substrate better, and the bacteria we're trying to keep out, try to keep it out by adapting the substrate itself. I'm going to talk a little bit about the progress we've done so far in identifying soils. So where these soils came from, the soil characterization, and then the disease screening tests we've done so far with a little bit of preliminary data, which I think is really interesting. So we've identified 2 farms that the University of Sheffield has had some research previously published showing that they were able to suppress disease. They also have really well documented history and farming practices, so we can hopefully figure out why that microbiome is suppressing disease. We've collected these 12 soils, and then we've developed a rapid disease screening process, which we've been using a different pathosystem. So we've been using Pseudomonas syringae, which has had a bioluminescence transformation. So it basically shows bioluminescence. And the plan is to grow tomatoes and then challenge the tomatoes with the Pseudomonas syringae, measure the microbial communities, freeze them, and then store them for isolation at a later stage. And what I really wanted to show is we've been doing these pairwise comparative tests of some of the soils. And what we have identified so far from bioluminescence data is a big difference in 2 margin soils from one of our farms, one with the conventional farming practices and the other one sustainable practices. In terms of sustainability for this test, it's a more diverse cover crop that's been sown in this margin soil. And we did it in 2 different doses of Pseudomonas syringae. In the low dose, we saw a difference between conventional and sustainable. And this was even more noticeable in a high dose challenge of the pathogen. And we suspect this is due to ISR, but we haven't confirmed that yet. That's going to be part of our next work. And we're also not sure, obviously at this stage, whether we're going to be able to transfer any of this microbiome into a hydroponic system, and whether it will still display this disease suppression. So we're first going to screen that again in a hydroponic system, but with Pseudomonas syringae before we move on to our Rhizobium Radiobacter and see if it also works for that. And with that, I'm just going to hand over back to Jake.
Okay, thanks, Harry. Yeah, so my stream that I've been working on has been focusing on the Radiobacter. So the first thing we did was we've gathered a range of isolates that have been predominantly grown with tomatoes, a couple with cucumber, and some of these isolates with or without the the root-inducing plasmid. The second part of my work so far is to build a hydroponic pathosystem, and within this I've done a very generalized run, which I've compared root-inducing plasmid isolates with non-root-inducing, and Pseudomonas, for instance, is a negative control. And then one thing I've explored is the wounded and non-wounded root treatments. This is to determine whether or not direct wounding has an effect on growth. And then I've been developing a drench analysis for opening detection. So going back to whether or not we can detect infection before we're starting to see any symptoms, and doing it in a fast and hopefully very precise manner. And to do that, I was using rockwool blanks with dosages of opiums applied. And then probably the most important thing for me in this part of work has been to understand the Radiobacter's growth and growth conditions, because it is a challenging bacteria, by no means easy one to work with. So these are 8 isolates that we've got at the University of Sheffield, and the 3 with the stars next to the 3 that I put in the first run of the pathosystem. And this is just a quick idea of the sort of scale of it. So the picture on the left shows you the plants at the early stages. They're in a mineral wool that's suspended halfway up these pots to reduce splashing. The drip feeders that are used to feed them are covered in copper, which are both countermeasures to stop spread of the bacteria around the system. It's an open system as well, so it drains into a tank for disposal. It doesn't recirculate the water, or nutrient solution, I should say. And then the picture on the right kind of shows you my first mistake with my first run on the pathosystem, which is to try and get too many plants in at the same time. So the next run will require less plants, that's for sure. But we just harvested last week, so we haven't got a great deal of data at this stage, but some photos just to show you how things are going. So these are 4 control treatments. The 2 on the left, Pseudomonas non-wounded treatment, and a non-root-inducing plasmid wounded treatment. You can see they're both affected quite severely with the root mat disease. And then the 2 on the right don't appear to show any great root mat disease there, which indicates to me there's been a great deal of cross-contamination within our system, and that we kind of need to try and identify what the method of cross-contamination has been, which has made me question whether or not we have an insect vector within our greenhouses, and means we might need to actually move to a far more controlled environment set up on our next run. Some general observations at this stage is that the cross-contamination wasn't universal. It was there, but it wasn't universal. And as I said, there's a possible pest vector there, but this is something we'll look into in the next run. And one big thing was the effect of moisture. I generally run a fairly dry hydroponic system. And one thing I noted was that while I was drenching the mineral wool to sample for opines, you would see spread of the root mat disease much faster. And concurrently with that, you would see that root matting tended to be more densely occurring around where nutrients are introduced to the mineral wool or where they flow to. Still to come on that work, obviously I need to run qPCR analysis, and I'd like to try and build a geographic map of colonization within that system, just to understand where cross-contamination came in a bit better, well, to understand that a bit more clearly. We're looking to develop a mass spec technique for opine detection. We'll be using a metabolomics approach on this front, so hopefully we'll have quite a nice high precision and fast analysis system running there. And then I need to quantify the value of infection based on the AHDB scale. They have a 0 to 6 scale of infection, just to compare that to what we saw or what we hope to see in opines. Yeah, and then I think the next steps for us as a team will be to look to transfer the microbiome that we've found within what we hope is ISR soils into synthetic substrates, and see if we can have successful colonization within the hydroponic systems. We have had previous experience of successful colonization in aquaponic systems, but these are quite a different proposition altogether. We'd like to determine whether or not the ISR behavior is transferred into the substrate using rapid screening assays that we've already experience with so far. And then we'd like to identify communities in these disease suppressive soils using DNA sequencing as well. And I think that was everything.